Today I want to talk about something that I thought would be of interest to this group here. And that's all true. My computer is going to explode in about 3 seconds. I'm counting on it. What I want to talk about is that all rodents are not the same. And this is pretty cool. I love this picture, got it from the internet. This is a capybara, weighs about 80 kilograms, the largest rodent. This is the smallest rodent. It's a pygmy jerboa. It weighs about 2 grams. This is a little cladogram that I've drawn. I just wanted to show you the phylogenetic relationship of rodents to other animals. And it turns out modern studies put them very close to primates, closer, in fact, than cats. The different colors, the blue, the red, the yellow, indicate primary cortical fields. And the point of this is that there's a common plan of organization, cortical organization, that all species share, including rodents. But there are great differences even within a lineage. Rodents are highly diverse and prolific. They comprise 40% of all mammals on the planet Earth, 5 suborders, 34 families, and over 2,000 species. Despite this diversity, 40% of all studies of the brain and neural function are done on only 2 species of rodents, and those are rats and mice. And as I said, rodents vary in body morphology, sensory specialization, deal pattern, locomotion, lifestyle, and cortical organization. And this is just a little flavor. We have some rodents that glide, some that are arboreal, some that are semi-aquatic, and so on. So today what I would like to discuss is differences in cortical organization in the context of lifestyle. And I'm going to go kind of fast and do a pretty superficial job. I'm going to compare global features of cortical organization in several species of rodents. It's some comparative stuff we've just done in our own laboratory. I'm going to make some cellular comparisons of V1 in several species of rodent, and then just quickly overall visual organization. I'm going to do some quick comparisons of somatosensory cortex in a variety of different rodents where the data exists. And finally, I want to talk about how aspects of the cortical phenotype, some of this variation, is generated. This is a cladogram of rodents. This is the common ancestor of rodents. It was diurnal. This is a deal pattern where the orange represents diurnal species and the black nocturnal. And you can see what's really cool is that rodents vary in just a simple deal pattern, and that diurnality independently evolved in the rodent lineage 5 different times. We can take one family, Scuridae, and look at something completely different, like terrain niche, and we see, again, tremendous variability, where you have terrestrial animals, arboreal animals, cliff-dwelling, and so on. These are 2 examples of arboreal species, a flying squirrel, terrestrial African ground squirrels, and rock squirrels. And one can imagine that there is, in addition to this difference in lifestyle, there's a coevolution of sensory systems that allow them, and motor systems that allow them to live in these different environments. You can envision, and it has happened, the evolution of 2-cone color vision in arboreal squirrels, changes to the auditory system and the cortex associated with a semi-aquatic lifestyle or high altitudes, clearly differences in the motor system based on locomotion, where you're living, how you need to get about. So now I want to just focus, I just want to give you a little sense of the variability. And we can look at a variety of different aspects of lifestyle and see that type of variation across this lineage.
What's the last common ancestor was us, was human?
No, that's the common ancestor of rodents.
I don't understand. How far away is the last common ancestor was us?
About 70 million years ago. So if you go back, can I go back to the first slide really quickly? I can do everything quickly. OK, here we go. Actually, I can't do this quickly. So we're just going to have to, when I showed you that huge tree and I said, look, rodents are actually more closely related to primates than carnivores and insectivores and apotheria, and bats. So we're actually they're good models in some instances for primates because of the phylogenetic relationship. It's about a 70 million year difference before our common ancestors split. So I want to talk a little bit today about some of the comparisons we made of general overall cortex. And some of the species we looked at had different diurnal versus nocturnal patterns. So we looked at the tree squirrel, the ground squirrel, and the Nile grass rat, which are diurnal. The rat, Rattus norvegus, is nocturnal. Differences in arboreal versus terrestrial lifestyles. We have an arboreal tree squirrel, and then terrestrial ground squirrel, Nile grass rat, and Rattus norvegus, and differences in wild-caught versus laboratory-reared animals. So we had the squirrels were wild-caught. I actually talked to graduate student Katie Campy into going out and catching wild Rattus norvegus in Davis, so she did, which was very cool. And then we have some laboratory animals. The cool thing about some of these comparisons is these animals were caught in Davis, and they overlap In lifestyle, they overlap in the physical habitat. So some of them are living in trees, some of them are living on the ground, some of them are active during the day, some of them are active at night, and of course the difference between laboratory and wild caught. So these are really nice, I think, species to compare. And we compared gross cortical organization in terms of primary sensory area size, and I'm going to show you some cellular comparisons of visual cortex. And these are architectonic stains, rostrals to the right, medials to the top. They're flattened and parallel to the cortical surface, so you can get a good idea of the aerial distribution of cortical fields. Here's a Norway wild-caught rat, or laboratory rat, V1, big S1, auditory cortex. You can see in squirrels, this is California ground squirrel, have a huge V1, S1, A1. Most of these fields and most of these animals, or many of these fields, certainly the primary sensory areas, except for Nile grass rat, these architectonic fields have been linked or found to be coextensive with functional areas, V1, S1, A1, AAF, and so on and so forth. And we examined an entire series of sections, did this stereologically, and looked at the differences in primary cortical fields. Because if you look at this, you can say, well, geez, this guy has a huge cortex, and it looks like he's got a really big visual cortex, where this one has much smaller visual cortex. So we made these measurements and quantified them. And what we found was pretty interesting. And some parts of it were intuitive, and some parts weren't. So if we look at V1, the diurnal squirrels, and squirrels in general, had a significantly larger V1 than the rat groups. And if we just look at rats, the diurnal rat had a larger V1 than the nocturnal rats. The arboreal squirrel had a larger V1 than the terrestrial squirrel. And if we move to auditory cortex was larger in rats compared to squirrels. It was larger in the Nile grass rat than in the two rats, laboratory and wild caught. If we look at primary somatosensory cortex was larger in the nocturnal species than in the diurnal species. And it was larger in not surprisingly, the nocturnal rats versus the diurnal rats. But what's really cool, it's also larger in the laboratory rats versus the non-laboratory rats. And these are the same species. We see similar significant differences in the size of auditory cortex as well. So, and I'm going to talk a little bit now about some of the cellular differences. And these differences are not simply allometric. It's not just because brains have scaled up. Squirrels have a larger neocortex than a mouse or a rat. But if you plot log cortical area against, regress it against total neocortex, what we see is that different cortical fields change at different rates, and you have different slopes. So if you look at S1 in the rat groups, it has a very steep slope, and it increases more rapidly. If you look at V1 in the squirrel groups, it increases more rapidly. So I'm going to move to a different level of organization, and that is looking at cellular organization, neuronal and non-neuronal number, ratio, and density in the primary visual area of the wild-caught rats, laboratory rats, same species, and then the Nile grass rat, which is a laboratory rat. And we used the method that's now more commonly used, the isotropic fractionator method, in which you this doesn't look so good, unfortunately, but you can actually peel off the cortex, scrape away some of the myelin. You can backlight this flattened cortex, and actually you can see V1, S1, A1. You can dissect it out. You can make it isotropic. And so you have to squish all the cells together. And then you get samples based of some volume. And you can stain for all the nuclei, and then you can stain with nu-n for just neurons. And in this way, you can look at number of neurons per unit area. You can look at the number of neurons compared to all the cells that were counted and so on. You can use flow cytometry. You can do this the old-fashioned way with classic stereologic counting methods. But I'm not going to go through that. I'll talk to you later afterwards if you are interested in it. But there are a lot of publications on this. And so what we looked at is the Nile grass rat, which is a lab rat, laboratory Rattus norvegus, and then the wild-caught rat. And just, there are lots of different things, but let me just give you the flavor of what we found. If you look at wild-caught and laboratory rats, same species, the wild-caught rat had a significantly larger percentage of neurons in V1 than the laboratory rat. If you look at neuronal density, the wild-caught animal in V1 had significantly larger, greater neuronal density than in the two laboratory animals, despite the fact that this is a diurnal animal. And if you look at measures that have been recently described, like the somatic neuronal quotient and the encephalic neuronal quotient, what you find is that the wild-caught animals far exceed what would be expected for an animal with a brain that size. So in terms of just gross aspects of organization, you're probably going to hear more about this from other people. These are just studies that I've redrawn from other people's work, some of it from our own. This is a California ground squirrel, tree squirrel sort of combo. This is a mouse. This is work from Draeger, Wager. I saw Mike Stryker. He's probably going to talk about his optical imaging stuff. This is a rat. To Blakemore, what I try to do is just have mainly functional descriptions of visual cortex. And what you can see, so there's some variability. There's no question there's some variability in the same species due to just technical differences or how you want to subdivide the cortex and so on and so forth. But there are some really clear differences in visual cortex in terms of organization. And if you do a larger comparative analysis, what you see, and you saw this in the first slide, is that if you look at these squirrels, these diurnal squirrels, they have a large amount of cortex devoted to visual processing. So if you want to break it into sensory domain allocation, a large amount is processing visual information. And as I said, this is not simply due to an increase in the size of the cortical sheet. You have the size of homologous cortical fields is different in some species. So V1 is actually relatively larger if you look at it relative to the size of the cortical sheet in diurnal species, as I've showed you earlier, than in something like a naked mole rat or a mouse. The number of cortical fields have been altered, and I haven't talked about that, but connections, cortical and thalamocortical. So if we move on quickly to somatosensory, I'd love to talk about auditory and motor cortex, but I don't have enough time, and I'm not going to talk a whole lot about somatosensory cortex. I just, like I said, want to give you the feel of it. So of course, we all are familiar with the rat primary somatosensory cortex with its granular and disgranular zones, and its really beautiful relationship to architectonically defined regions of cortex, whether it's Nissl stains, cytochrome oxidase, and so on. But the study by Chapin and Lin pointed out a couple of important things. One is that the responses of neurons in S1 are not homogeneous. Neurons in granular cortex generally respond to cutaneous stimulation. Neurons in disgranular cortex, this large region right here, respond in awake animals to stimulation of deep receptors, and in the perigranular zones to deep and cutaneous. There's also a transitional zone right here. Recently, well, for quite some time, people have been looking at differences in connectivity between granular and disgranular, and there's a nice study that just came out in JCN looking at subcortical connections and shows that the cortical connections of these fields are different. The thing about rodents, and this is nice, is that beautiful comparative study by Woolsey back in 1975, and what he showed is that barrel cortex is not ubiquitous across rodents. Some rodents have barrels. Most squirrels have really minimal barrels or no barrels. This is a squirrel family. Some kind of have barrels. And what they also noted was that the presence of barrels is not associated with the presence of whisking. So some animals whisk and don't have barrels, and some animals have barrels and they don't whisk. They further said that barrel formation appears to be related to brain size. So if we look at other rodents, just now general somatosensory organization, they don't look like rats and mice. They look quite different. So here's a rat. You have a huge amount of cortex devoted to the vibrissae, forelimb, hind limb, trunk, rest of the face. This is a grasshopper mouse. You have a lot of cortex devoted to the vibrissae. But if you look at something like the gray squirrel, you do have a lot of cortex devoted to the face, but it's devoted to the lips, the buccal pads, and the teeth and tongue. They store food in their mouth. They use their mouth as a tactile discriminator. If you look at something like the naked mole rat, this is work by Ken Catania's group, a relatively small vibrissae representation, very large buccal pad, and also upper incisors and lower incisors, which move independently, which they use to discriminate objects. So all rodents appear to have specializations of the face compared to primates that have really specialized hands and lips and tongue, but it's not necessarily the vibrissae. And if we look across somatosensory cortex, we see changes in the size of homologous cortical fields. We know that in rats it's relatively larger than in diurnal squirrels. The extent of modularity or interdigitation. So this sort of really beautiful relationship between granular and disgranular zone or perigranular zone, where they're completely interdigitated, is not seen in other species. We see sometimes partial invaginations in species, which may be the same homologous field that's just organized quite differently. And in other species, we don't even seem to see that, particularly smaller brains. There are differences in connections and differences in number of fields. So the common ancestor of rodents probably didn't look like the specialized rat or mouse. It absolutely had a primary visual cortex, primary auditory cortex, and a primary somatosensory cortex, which may or may not have been segregated into a dysgranular zone or something like a R or 3A. It may have had an auditory belt and a little bit of visual cortex. This led into, in some lineages, you had the nocturnality was maintained. You have animals that slightly increase the size of the cortical sheet. They expanded somatosensory cortex, specialized and integrated sensory and motor barrel systems. In other lineages, you have a very, very large cortical sheet. You have the evolution of diurnality. You have expanded visual cortex, more and larger motor areas, multiple somatosensory areas. And in some species, you actually got a miniaturization of cortex, like in the subterranean mole rat, their brains were probably likely a bit smaller than the common ancestor. Miniaturized visual cortex, auditory cortex, a very, very large S1 that's devoted to specialized receptors, such as the incisor and buccal system. So comparative studies, we examine the products of what evolution produces. They tell us what sorts of changes evolution has produced. But the problem is we're just examining frozen moments in the moving picture of that lineage's life. So these changes are really quite dynamic in species over time and within the life of an individual. Developmental studies allow us to uncover the mechanisms that generate aspects of the cortical phenotype, so how these changes emerge. So just really quickly, the last few slides. I'm doing OK on time? Oh, I have 5 more minutes? Oh, OK. What factors contribute to the variation across and within species? And of course, is it genetic or is it activity dependent? So I'm going to look at one aspect of cortical organization, which is changes in cortical field size. So how is the cortex increased in size? Well, evolution has produced larger animals with larger brains. So I'm not saying that allometry doesn't exist. You scale up your body, you scale up your brain. But usually what happens, this is a comparative study by Campos and Welker. This is a guinea pig, this is a capybara. These are drawn to scale. The capybara has an absolutely larger brain. It has an absolutely larger body. And it looks like, and this probably is V1 plus V2, it looks like fields are basically scaling up. So this does happen. But evolution has produced animals with proportionally larger neocortex and more parts, like the squirrel. So this brain isn't just a scaled up version of something like this, but it has more cortical fields, more differential expansion of some sensory systems, and so on. So increasing the size of the cortical sheet is necessary, but may not be sufficient to increase the number of cortical fields. And there have been a lot of studies. This isn't anything that my laboratory does. There are several possible intrinsic genetic mechanisms to increase the size of the cortical sheet. I'm sure most people are familiar with Kriegstein's work and Nochter's work, where they suggest that species are increasing the number of progenital cells. There's the work of Chen and Walsh looking at beta-catenin and the number of times cells re-enter the cycle. The important point is that some alteration in cell cycle kinetics takes place, probably genetically mediated. However, it may be different. What's changing may be different in different animals, although the phenotype, a larger cortical sheet, is the same. OK, genes intrinsic to the neocortex alter cortical field size. This is work from the O'Leary lab, where he overexpressed EMX2. Sorry about this, but I don't know why they draw their brains up in this weird orientation. This is rostral, because, of course, the correct orientation is my orientation. This is medial. Here's V1 in a normal animal. And you can see, by altering the expression, overexpressing EMX2, we can greatly increase the size of V1. We can delete CoopTF1, which is expressed caudally in cortex. And what we see is somatosensory cortex, here's the barrels, are smushed all the way back. V1 is still here. Auditory cortex is now smushed all the way back. And if you make an injection of an anatomical tracer here, you're getting projections from the ventroposterior nucleus. So the lamocortical afferents have also shifted. OK, so when we tend to think about changes in the cortex, we don't always think about the animal as a whole. We don't think about the body. There are genes that are extrinsic to the cortex, but intrinsic to the animal, that can be altering aspects of peripheral morphology. This is work that's done by Kritos and Chen. And they look at the hand phenotype of a bat versus the forepaw phenotype of a mouse. And they compared expression patterns of Hox genes involved in early development of the forelimb. And they found slight differences at middle stages. And read the stuff, they have beautiful detail on this, that can account for these large morphological changes. In turn, if we look at the cortical organization of a mouse versus a bat, we have a relatively small forelimb. And if you look at the bat forelimb, this includes megacoropteran bats and microcoropteran bats, you have a very large expanded representation of the forelimb. And they have evolved specialized touch domes that are exquisitely sensitive to small changes in air pressure. which would make sense if you're flying and there's a big wind. So specializations are associated with peripheral morphology, and we see this not just in our classic vibrissi rodent example, but also this is work from Catania Lab in the star-nosed mole, in the naked mole rat, and so on. And so do genes that encode aspects of cortical organization, or it seems as if, they must covary with those that encode aspects of body morphology. And of course, body morphology is always associated. Specialized body morphology is associated with specialized use. And specialized body morphology, by that I also mean specialized sensory receptor arrays, evolution of new types of receptors, changes in density of receptors, and so on and so forth. So changes in the relative activity from different sensory systems can also alter cortical field size. This is actually from Monodelphis domestica, where we did early bilateral nucleations before thalamocortical afferents were established and before retinal ganglion cells had grown into the diencephalon. This is rostral, this is medial. These are normal animals. Here's primary visual cortex V1, area 17 in normal animals. And in these animals that had bilateral enucleations, what we found is the overall size of the cortical sheet was not changed, but the size of V1 had gotten very small. Now, this isn't visual cortex, because they have no eyes, but it's defined architectonically as area 17. And so what we found was V1 was significantly smaller, and S1 had increased in size. But there were no change in the size of the cortical sheet. So you've changed the relative activity of different sensory systems. You've changed the pattern of activity by removing one sensory system. So now it's getting a lot of somatosensory and auditory. And it turns out that all of this cortex that's supposed to be visual is taken over by the auditory and somatosensory system, both functionally, and if you look at connections, you'll also see multimodal connections. OK, so I'm just about done. Ways in which the phenotype can be altered. And the point of this talk is that, I'm going to talk, I showed one example, cortical field size. There are multiple ways in which some aspect of the phenotype can be altered. You can simply change the size of the cortical sheet and increase, so it's just a simple allometric change. You can alter genes intrinsic to the developing neocortex and change the size of a cortical field and its location. You can alter genes intrinsic to the developing body but extrinsic to the cortex, which is related and changes the size of cortical fields. Alterations in the sensory receptor array or activity or use of that particular part. You can alter the physical environment in which the animal evolves. And there's some nice work in Fitzpatrick labs and other laboratories, and we know this from the visual system, that if you change the conditions under which an animal is reared, you can change ocular dominance, you can change direction selectivity, and so on and so forth. And of course, then there's adult plasticity. So there isn't a single way, and different rodents and different mammals are probably utilizing, or their brains are changing in a variety of different ways, depending on their genotype, but also the environment in which they're unraveling. So genetic alterations and alterations in sensory-driven activity drive change. And both genes, or genetic cascades, or the way they're deployed, and the laws of physics place formidable constraints on evolution of the brain and restrict the avenues of change. Systems-level changes to the phenotype are limited, and they're predictable. You can name maybe 10. And maybe there's more, but I suspect that it's very similar at a molecular level. The persistence of a common plan, even in the absence of use, and convergent evolution of similar features or homoplasy at all levels of organization provides support for this. And one of my favorite examples is the barrel field. But this isn't a rat. This is actually a brush-tailed opossum. And if we look at where the brush-tailed opossum, oops, sorry. Uh-oh. If we look at the phylogeny or cladogram, and we look at the presence of barrels, this is also ocular dominance columns, the brush-tailed possum is a marsupial. No other marsupials appear to have barrels. And if we look at rodents, rodents have barrels, but not all rodents have barrels. So this is the independent evolution of this thing. And I think the fact that these things look so remarkably similar is not a coincidence, but clearly underscores that the constraints imposed on evolution. And if we can look at ocular dominance columns again and look at that they've independently arose in different lineages. This is an old tree. This is from an old paper, so the relationships aren't completely correct, but they're close enough. Interestingly, the work by Horton in squirrel monkeys shows that even in the same species, you can have the presence or absence of fields. So I'm done. I just want to introduce my laboratory. Katie Campy did all the comparisons on rodents, and it's mainly her work that I showed. Adele Selke's doing some of the isotropic fractionation, and these guys are just doing other things. Important things. OK, so I'm done.